Hey class, so picking up where we left off, we were talking about enzymes, as you can see here on this slide. So we're talking about what affects enzyme activity. So does anyone have any examples? Yes, you in the back. Okay, wrong. Okay, 3 factors that affect enzyme activity. Number 1, environmental conditions. So this includes things like heat and acid levels and things like that that can maybe deneutralize an enzyme, because you have to keep in mind enzymes are proteins that are balled up amino acids. They can unravel. Cofactors and coenzymes, and then enzyme inhibitors, which, are things that kind of attach to the enzyme and then affect the way that other things shouldn't attach to the enzyme. So, continuing on that note, environmental conditions. Extreme temperature, like I said earlier, are the most dangerous ones. And then you have high temps may denature or unfold the enzyme. This is very redundant since I already explained things perfectly earlier. pH, and then ionic concentration, so like salts and such. Then cofactors and coenzymes. So inorganic substances like metals such as zinc and iron and vitamins are sometimes need for proper enzymatic activity, sometimes need for fix later, note to self. And an example of this would be iron must be present in a quaternary structure, hemoglobin, in order for it to pick up oxygen. So 2 examples of enzyme inhibitors, I believe they would be a competitive inhibitor, which are chemicals that resemble an enzyme's normal substrate and complete. Do you have a question in the back? Front row? Sorry, what a troll, am I right? And compete for its active. So think about if you have someone who's the starting spot and you want to be the starting spot, you're kind of like the competitive inhibitor for them on the field. And here's a little graphic to help you learn a little better, because visual learning is just as important as audible and reading learning. Okay, and then you have the non-competitive, which non-competitive inhibitors are inhibitors that do not enter the active site, but bind to other parts of the enzyme, causing the enzyme to change shape, which in turn alters the active site. So, examples of these would be this would be a competitive, this is the active site, and this is something. This would be an example of a competitive inhibitor, because it is going directly into the active site. But if you had something like, this, it goes into, not the active site, but it still affects everything that's going on here would be the active site. So that would be non-competitive, because it's not competing for the spot of the active site, but it's still messing stuff up. And again, an example. It changed. You can see it. And so, we're done, class! Yay! I think I did good. Can we watch it?
Section.
It's still recording. It's still recording.
I pressed pause.
Hit this.
Wait, changing multiple?